Hello everyone, thank you so much for tuning in. So we're going to be continuing our discussion from my class. These are just going to be an overview of the notes from the slides so I could go back and study them later. Last time we looked at physical means of controlling microbes. This time we're going to be looking at the chemical side of things, so using different disinfectants that aren't physical, so we're not using heat or radiation or anything like that. This time we're going to be actually using reagents as well as chemicals that usually you would think of as disinfectants to destroy these microbes. Alright, so once again we're going to be going over some definitions. So we have sterilant, and this destroys all microbes. And they're also called sporicides. So again, we talked about those endospores. They're also known as sporicides. So they do get rid of those resistant endospores. And these are going to be used on critical instruments. So in a surgical setting or a medical field, the critical instruments are those that actually come into contact with the organs or the tissues of the body. So we're going to make sure we use a sterilant to get rid of all those microbes because we don't want to contaminate anything that is going to be making direct contact with those very vital areas. Next, we have high-level disinfectant. Remember, disinfectant does not get rid of all the microbes. There are some that are still left afterwards, but we're getting rid of as much as possible. So specifically, the high level will get rid of viruses and vegetative cells. So a vegetative cell is one that is able to be metabolically active as well as divide. It's different from our endospores, which would be gotten rid of by the sterilant. The disinfectant only works on the metabolically active cells. And these are used on semi-critical instruments. Alright, now we have low level and intermediate. So actually, let's go in order. Let's do intermediate next. Another type of disinfectant, and these will destroy vegetative bacteria, fungi, and most viruses. Keyword there being most again. These are going to be on non-critical instruments. So again, non-critical instruments, these aren't coming into direct contact with the living tissues during a medical situation. So we don't have to use a sterilant on them, but we could just use an intermediate level. Now let's talk about the low-level disinfectants. These will destroy vegetative bacteria, fungi, and enveloped viruses. These are going to be used on floors and walls. So not on instruments themselves, but just on the surroundings. And again, enveloped viruses, so ones that do have that lipid or protein coat, not the viruses that lack a coat. Those are going to be tougher to deal with, as we've discussed up here. Non-enveloped viruses, so they resist disinfectant. All right, let's move on to our first chemical that we're going to be looking at, which is alcohol. Then ROH, of course, that's in organic chemistry. The mode of action, or the MOA, this is how it's going to stop the microbes. What's its method of stopping them? It's going to coagulate proteins and damage lipids, specifically the membranes. And then usually you'll use one that's 60 to 80% in an aqueous solution. So AQ, and it's usually ethyl or isopropyl. And they get rid of vegetative bacteria, so bact, and fungi. No effect on those resistant endospores, as well as naked viruses, or viruses that aren't enveloped in anything. So naked viruses, and those are extremely resistant, so alcohols aren't going to affect them. So this is used as both an antiseptic as well as a disinfectant. Alright, so from last video when you're going over physical means, antiseptic means it's a chemical or a method that's going to be used on living tissues. And then disinfectant is for inanimate objects. Okay, so limitations include a quick evaporation, and this is going to decrease the contact time. It won't be able to reach everything because of how quick alcohol is, it evaporates or dissipates. And it damages rubber and plastic. So the pros are all of these, where it gets rid of the vegetative bacteria, fungi. It's able to be used on living and non-living, but here are cons in the quick evaporation time as well as the damage to certain material. Alright, next reagent on our list is the aldehyde. The mode of action, EMOA, it inactivates proteins and nucleic acids. So we'll just simplify it, say DNA, but it does say nucleic acid, so technically it's DNA, RNA. Alright, so one type that is commonly used, let's just highlight some of the main things here. Okay, so one of the main types is glutaraldehyde. And this is a sterilant. Remember, up here we learned that a sterilant destroys all microbes. So this is extremely powerful. However, it's toxic. Even though it's a sterilant when things are immersed in it for 12 hours. So we have kind of a double-edged sword because there's that danger and that toxicity. We also have formaldehyde. This is that preservative for taxidermy. So it's used as a gas or formalin. A formalin is a 37% solution. And we could kill bacteria, but we only inactivate viruses. And this is used for vaccines. So there's an inactive virus in certain vaccines, and that's allowing for the immune system to recognize it, get used to it, so you could fight it off and to recognize that particular virus if it ever gets into your system after the vaccine. And again, this is preservation. Alright, let's go to, this is an interesting one. I've never heard this type before, other than this class. It's a biguanides. This section is called biguanides, and I sound like Benedict Cumberbatch when he's trying to say penguin. He kept saying penguin, I do not know how to actually pronounce this. This is the first time I'm actually learning about biguanines. The mode of action is you are able to bind to the cell wall and the membrane. One of the most common types is chlorhexidine, I think. All right, and low toxicity. So it's much safer than our glutaraldehyde. It destroys vegetative bacteria, fungi, and some enveloped viruses. It's a skin. I don't know why I was spelling skin, sorry. Antiseptic. Pre-surgery. It's also seen in mouthwash. And here's a funny story about mouthwash. So we talked about Lister earlier. So Listerine does come from Joseph Lister. It's named after him because he was talking about creating an antiseptic method of surgery where Listerine is a mouthwash, so it's an antiseptic for your mouth. That's the connection there. Pretty interesting. Listerine was named after him. All right, ethylene oxide gas. This is our next substance. And it's a sterilant. Remember, that gets rid of all microbes. It's in a chamber similar to an autoclave, and the mode of action is reaction with proteins. And this is bad news, of course, for the cells, because we all need proteins, even bacteria, and all those microbes, of course, need proteins. Getting rid of them is really unfortunate for them because they can't use the proteins for function. It's able to be used, the ethylene gas. Or ethylene oxide gas. It's used to sterilize heat and moisture sensitive items. Such as fabrics, maybe equipment, and then lab items such as petri dishes are also sterilized using ethylene oxide. Again, it's just like an autoclave, but you're using this certain gas in order to get rid of everything because it's reacting with proteins. All right, let's talk about halogens. Right, so the mode of action destroys and oxidizes proteins as well as components of the cell. All right, the biggest one here, chlorine. Not chlorine gas, that's toxic. Do not make chlorine gas, please. You're going to be in a very messed up spot. That's really dangerous stuff. All right, so this destroys all microbes. And even viruses, all microbes including viruses. This is a disinfectant. So, if it kills all microbes, you might be wondering, wait, why is it called a disinfectant if it kills all? Well, there are some species of bacteria that actually live in chlorine. They're able to metabolize it. They're able to use it. There's a lot of chlorine in drinking water, which we've talked about the treatment of water in the previous video. This species, Crypto, not the super dog, but Cryptosporidium, as well as the cysts of Giardia, these are also genera, so I'm going to underline them. I can't really write in italics. these cysts will actually survive. That's why it's still a disinfectant. Even though it kills all microbes, it's a disinfectant because of these two species are just able to utilize the chlorine in water. That's why it's very important to regulate drinking water, make sure it's clean. All right, and organic compounds sometimes interfere with chlorine. They kind of make reactions. We talked about those byproducts last time too when we were talking about treating drinking water for microbes. Another form is chlorine dioxide, and this specifically is a disinfectant and a sterilant. That's all we need to know on that one. Another larger one is iodine or iodine. This kills vegetative cells, but not endospores. So again, the endospore is extremely resistant, already a large issue. Iodine won't work on it. This is an iodophore actually. Man, I'm just messing up the pronunciation on all these things. This allows for a slow release from a carrier molecule. Pseudomonas actually can survive in iodine stock solution. So again, it's a disinfectant. It doesn't get rid of everything. Very tricky, this one. Very resistant. Alright, now let's talk about metal compounds. This might have been a surprise attack, a sucker punch. You weren't expecting metals to be in this. Alrighty. So, mode of action. They combine with sulfhydryl groups of proteins, and this leads to denaturation. Okay, so silver. It's one of these metals. It's an antiseptic. Therefore, it's used on living tissues. Silver nitrate, so plus nitrate. These are those eye drops given at birth to stop the bacteria the genus Neisseria. Gonorrhea. So when babies are born, nowadays they just use an antibiotic, but before they would give silver in eye drops to babies to stop them from having gonorrhea. And we also have compounds of mercury, tin, copper, and other trace metals that are used as preservatives. And these prevent microbes from growing in water. Specifically, water left in those tanks in all those facilities. Alrighty. Now let's talk again about a gas, ozone. So O3 or ozone. Not 007, just O3. Alright, so this is the mode of action that's going to be an unstable form of regular O2, and it's just going to oxidize anything that comes into contact with it. So extreme oxidation, because it's literally O3. It's very reactive. So that's also a lot of explanation points, because it's a very, very angry molecule. It reacts with everything. There it is, like shooting all these other molecules in and oxidizing them. It decomposes quickly. And it's used an alternative to chlorine for drinking water. It's a disinfectant specifically. Let's talk about peroxygens. These might seem a little bit more familiar. The mode of action is to oxidize cellular components. And of course, this would make them sterilants. And fun thing about these, they're less toxic. They're biodegradable. And there's no residue. Alright, so main one we're going to be focusing on is, of course, H2O2, hydrogen peroxide. And it depends on the surface. So surface dependency. It breaks down into O2 and H2O. Those aren't very harmful. It's most effective on inanimates. It's best to use hydrogen peroxide on non-living objects. We also have peracetic acid. Never heard of this one. This is basically more potent. And this one you can use as something that would clean off living tissues. I know I'm scrolling up. There we go. We also have phenolics. So we were talking about the carbolic acid that Joseph Lister was using when he was trying to make surgery safer in his time. So now we're going into that section again. Phenolics. And back to organic chemistry, a phenol is a benzene ring, or it looks like it has a benzene ring. This is an aromatic ring. The hydrogen at position 1 is replaced by an OH. The mode of action would be to destroy the cytoplasmic membrane, as well as denature proteins. Lister again, and carbolic acid. This substance has an unpleasant odor. It's also an irritant. So they have this new version, triclosan, that's being used. Less irritation. And it's used in antibacterial soap. It's much more of a pleasant substance. This kills most again being the key word, vegetative bacteria. It doesn't work on viruses. So this would be a disinfectant, not a sterilant. It remains effective even when there are other detergents and contaminants. However, it leaves this really good antimicrobial residue. So unlike alcohols that just evaporate quickly, there's a residue from the phenols that will just continue to act as an antimicrobial for you. We also have something called a quaternary ammonium compound, or a quat. The mode of action for a quat is a catatonic detergent, cationic. So this will react with the membrane. Low toxicity, and it's used in food prep, specifically disinfection. Removes dirt, organic material, as well as organisms. Stops, or I should say kill, not stops. It doesn't inhibit, it just destroys vegetative bacteria and enveloped viruses. Not all viruses, just those that have an envelope. It doesn't work on endospores, mycobacteria. Again, mycobacteria do not have a cell wall that has peptidoglycan in it. Sorry, I'm getting a brain fart. All these compounds and stuff. Mycobacteria have this waxy cell wall, so it doesn't affect them. Non-enveloped viruses aren't also affected. Pseudomonas are resistant. And just like iodine, the Pseudomonas will grow in these quaternary ammonium compounds as well. Okay, so we talked about all those reagents, so let's talk about hospital settings. So why do we need to, of course, control microbes in hospitals? There's a danger of healthcare associated infections. Again, we're just swapping gears here, switching gears back to definitions and bigger picture concepts instead of looking at the specific reagents. So, nosocomial infections, or HAIs, these are going to be affecting those in hospitals and other medical offices like that, since patients are much more susceptible to infection. And because of all these invasive processes, such as surgery, that is opening the body and all the very sensitive and vital organs to the potential of being infected by outside bacteria. And pathogens, again, are common in hospitals. There are tons of pathogens at hospitals. You'll see feces samples, urine, other bodily fluids. And it's best to avoid respiratory droplets. Remember, during COVID in 2020, 2021, and even now, because we're seeing a new COVID variant as I'm making this video, there's a new one they just rolled out vaccines for a few weeks ago. The danger was like the 6 feet thing because of the respiratory particles of just speaking to someone. Of course, that also spreads other viruses and bacteria too, but COVID, one of its main things is a respiratory setting. So those respiratory droplets, of course, are going to be a potential for getting these pathogens, as well as other secretions too. Sterilize instruments. And people, of course, we need to sterilize instruments. Yes, this is the main point. We need to avoid introducing infection to deep tissues. So Okay, so we still have that concern of prions. We talked about these tricky buggers before. You really need high temperatures. You can't just autoclave. The sterilants will work on everything except for prions. Even by sterilants, so they can't be easily destroyed. To use a lot of strong heat and stuff like that. Alrighty. That covers the methods of microbial growth control, as well as an overview of sterilization, disinfectant, and the processes of going into antimicrobials and other ways of cleaning all these little things up. Thanks so much for tuning in. This is part 2 of the chapter that we went over in class. Part 1 was released a few days ago, so please check that out too. And have a good one. Please do something nice for someone. Take care, everyone.